Hello, this will be lecture number 7 in a series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we will be discussing amino acids, peptides, proteins, and this will be a very interesting topic to go over. We talk about proteins as a number of amino acids as being their constituent points. And we talk about oligomers and polymers, but proteins are different in terms of the size and complexity, and they can exist in a variety of different configurations and different formulas. There are 20 amino acids in all species, and the amino acids are the constituent building block of the protein. And the proteins are, by definition, a polymer of amino acids. All of them are in alpha structure. There's another one that we'll be discussing a little bit later on. They consist of a carboxyl group and amino group. They're covalently bonded to an alpha carbon, and the formation of that carboxyl group and amino group forms what's called a peptide bond. And the R group on this is a side chain, and the side chain varies. It can be a protein, as you would see in glycine, or it can be a highly complex molecule. It can be aromatic, it can be basic, it can be acidic. There's one amino acid that actually does not have a peptide bond that consists of a frozen bond. We'll be discussing that. Each of these amino acids has a 3-letter symbol or a 3-letter abbreviation, and we use a 1-letter symbol in bioinformatics, and I'll be showing that very shortly, at least the exceptions, to make it easy. The alpha carbon is a chiral center. There, again, we discuss chiral carbons. They have 4 different constituent groups, and these are all L. All these are L. Racemic mixture is L and D, and because these synthetic enzymes, synthetic biomolecules represent and recognize asymmetric structures, they'll respond to one configuration, not the other. So all the amino acids are of L. And if you want, you can also use the RS system, where R is clockwise and S is counterclockwise. But again, amino acids are exclusively L stereoisomers, and these chemical reactions are stereospecific. So again, the amino acid nomenclature is determined by the R group. And what does that look like? The R group is a shaded part that we're seeing. So to get these in organized capacity, we have the nonpolar aliphatic R groups. There's glycine, which is just a proton. This is the peptide formation is going to be the reaction between the amine group here and this carbonyl group with elimination of water, and that causes the peptide bond. We have alanine, which is a methyl group. We have proline, and proline is the exception to this, where instead of a free amine group, we actually have an amide group that's attached, and you form a 5-ring structure with 2 methylene groups, or 3 methylene groups here. This is the one that does not really have a rotatable peptide bond. There's also valine, which is kind of nice because it's a V shape here with this constituent. Leucine adds another methylene group to this link. You have isoleucine, which has one of the methylene groups is replaced by a methyl group here, and methionine, which has an ester with a sulfur at this point. You also have polar uncharged R groups. So these are polar groups, but they do not form acids or bases. So you've got serine with a hydroxyl group at the end, threonine with 2 carbons in the R group with a hydroxyl group at the penultimate carbon, cysteine with an E there, which has a sulfhydryl group and the methylene group here, asparagine, which you'll often see in certain amino acid sequences with the amine group here, carbonyl, methylene, and then glutamine, which is the same, although there's now an additional methylene group. We have the aromatic category up here. We have phenylalanine right here, tyrosine, which has a hydroxyl group at the distal carbon, and tryptophan, which is a little bit more complicated, but it's a bi-ringed structure here with an aromatic characteristic throughout. Then there are three positively, the three amino acids with positively charged R groups: lysine, which has an amino at the end; arginine, which has an amino at the end, but another amino here and amine here. This is sort of fascinating to show the vast array of reactions that carbon can undergo. Histidine, which is a five-ring carbon aromatic here. And then finally, we have negatively charged R groups, and aspartate, which has a carboxyl group at the end, and glutamate, which has an additional methylene group. So some of these differ. If you just study these, you don't try to memorize necessarily the words, but just try to think through the structures, the difference between these. There is some method to the madness. If you can organize the grouping, that would be very useful. I mentioned the bioinformatics. In large measure, the single letter is the first letter of the word. So glycine, you're looking at basically for the three amino acids, you just take the first three letters. But for bioinformatics, we're using a single letter. You just take that first letter. So glycine is G, alanine is A, proline is P. But the exceptions are what is critical. So the exceptions are phenylalanine, where the single letter is F, which I guess it sounds like pH. Tyrosine is Y, which is the second letter in the word. Tryptophan is W for tryptophan. Asparagine, which is down here, is N. Glutamine is Q. And lysine over here is K. And aspartate is D, and glutamate is E. So those are the exceptions for the bioinformatics role if you want to use and if you want to go to that. Proteins have a fascinating chemistry all their own, and we talk about the protein absorption of tryptophan and tyrosine at 280 nanometers. And what's interesting about that is if you're attempting to isolate protein from any sort of purification process, the 280 nanometer is a good way to use a general spectrum to determine at what point in your elution of a solution containing a various protein, whether it's a large one, a small one, the one that you're interested in. 280 is where you set the spectrometer to determine if you're getting protein out of a particular medium. And this is how you can also use the absorbance for protein quantitation, because the magnitude of that absorbance is going to be proportional to the concentration. So the Lambert-Beer law is used, and the absorbance that you're seeing is going to be equivalent to the log of the concentration or the incident intensity, I should say, and then the transmitted light. So when you look, and this is equivalent also to ECL, molar extinction coefficient is E, C is concentrations, and concentration and absorbance are proportional, and the length of the absorbent medium. So when you have light shining through a monochromometer, you get incident light of a specific wavelength, 280 nanometers, a sample cuvette with a certain length I, the length which is right here, length of the absorbent medium is L, I should say. The transmitted light is I, so incident light is absorption. The transmitted light is occurring here, and then the ratio of that is the number that you get here. So this is a mathematical construct, and you can use it quite nicely to get concentrations. we talk about uncommon amino acids, and these are seen in the urea cycle and biosynthesis of arginine. They're not, although all animals need the same 20 amino acids, we differ in the amounts of amino acids we can synthesize. So you can get derivatives of amino acids. We'll be covering some of that. But two of these are of particular interest. Ornithine and citrulline are present in the urea cycle, and they're also seen in the biosynthesis of arginine. This is not critical, but amino acids are weak acids and bases in solution. Is there enough of it to impact a physiologic condition? Probably less so than other salts that are present in solution in the organism. But you can use the amino acids, and going back to the structure, you can see that there were many opportunities, particularly with things like glutamine, to see several different places where there are charges. So you end up getting several pK values, each of which is contingent on a certain structure that is occurring. So you get you can titrate these with acids or bases to get Zwitter ion forms, which is where you get the alternate charges on the same amino acid. Basically, it cancels out the negative and positive charges at that point. And then on non-ionic form, if you can progress the titration to a given point. So when we look at non-ionic form, we have structures such as this. Again, R is a constituent group, and the amine and the carboxyls are forming the peptide bond as you get a protein form. The Zwitter ion is a construct where there's an internal shift, but the net charge is still zero. But you gain the negative removal of the proton from one of the oxygens here, and then the additional protons. So you get a trend to have the electronegativity go in this direction from here through here, drawing a positive charge here and a negative charge over there. So the Zwitter ion can act as an acid here, where you can remove the proton and get a free amine group with this, or you can use it as a base where you retain this, and then a proton is picked up over here. So you can have that sort of transformation occur. When you're looking at this structure, you can get, for example, an amino acid here, where you've got the low pH form at this point, where you've got no negative charge. A pK1 gets transformation from this form to here. Now you've got a net negative charge, and then we call that the PI, the isoelectric point, and then the pKa occurs when you get this sort of equilibrium here. Again, the pKa is going to be the product over the reactant, and you get two different buffer zones for glycine, which is your simplest amino acid. The second pKa, pK2 is 9.6, and the first one is 2.34. And this is the transformation between these two species here and these two species over here. By the time you get here, there's none of this. So we're looking at ratios in this range at this point. So again, these are all characteristic of the Henderson-Hasselbalch equation. And we've moved beyond water now. Now we're looking at things that are more physiologically interesting. So again, the net electric charge is zero. So the peptide bond is where we get the linkage of amino acids, and dehydration step forms that bond. Oligopeptide are few amino acids in linkage. Polypeptide are several amino acids in linkage. Proteins or polypeptides are over 10,000 Daltons. Dalton, again, is 1/10 the mass of a carbon atom. The nomenclature, again, has the amino terminus on the left and the carboxyl terminus on the right. So the free amino group in the construct is on the left side, and we put the carboxyl on the right with a string of amino acids in between. So we have the pKa value changing when it's part of a peptide, and the charge resides in the R moiety. Most of the proteins have below 2,000 amino acid residues. We have multimers, which are separate non-covalently bonded peptides. Some peptide proteins are linked covalently, others are not. One example of the latter is hemoglobin, which is a 4-unit. Many proteins have several subunits, which we'll be getting into shortly. Proteins have variability in amino acid residue components. They can have different conformations. Again, one dictate that I want to stress is that the structure dictates the form, and the form dictates the function. Conjugated proteins are proteins that are bonded to non-protein components. You can have lipoproteins, glycoproteins, and metalloproteins. So that'll be it for this talk. Thank you very much.